Good evening, everyone. So I hope I am audible and you can see the screen. So today we will discuss about enzymes. Okay. So we have different biomolecules like enzymes, proteins, carbohydrates, lipids, etc. So in that first we will discuss about what are the enzymes. So in this today's class, we will mainly concentrate on what is the chemistry of enzymes, classification, what is the mechanism of enzyme action, then enzyme kinetics, inhibition, activation, and specificity. So first we will discuss about enzyme chemistry. So what is an enzyme? So enzymes are biological catalysts. So if you remember, in any chemical reaction, usually we use catalyst. So what is the purpose of using catalyst? It will speed up the reaction. So when we take a reaction, in that we will have reactants, then two or more reactants, they react to form a product, one or two products. So this together called as a reaction. So this reaction can also happen spontaneously, but it will take long time, longer durations, maybe hours or weeks or days. So depends on the reaction. So here, why we use catalyst? It will speed up the reaction without involving in the reaction. So that is the important point. So it won't react with the substrate or it won't affect the product, right? So that is the main purpose of the catalyst. So same reaction, if it happens in a biological system, okay, because in our body every day, like many reactions takes place, metabolic reactions. So even there are reactions. So there, whatever the reactants we are telling, so here we name them as substrates. Okay, so these substrates, they react together to form product or products. So for to speeden up this reaction, we use, means we not voluntarily don't use, but there are some catalysts which are naturally present in our body, which are called biological catalysts. So enzymes are the ones which are performing this function. Okay, so they will speed up the rate of biochemical reaction. And these enzymes are made up of proteins. Okay, so we have, as I said, biomolecules, proteins, carbohydrates, lipids, like that. We have many biomolecules, and the enzymes are mainly proteins in nature. And these proteins, if we see, we have different levels of structures like primary, secondary, tertiary, and quaternary structures. So what are they? So primary structure is nothing but a group of amino acids. They arrange in a particular order to form a protein. So it's a linear structure. So that is the primary structure. Then this linear polymer of amino acids, it will form certain, it will assume some shapes. It will fold themselves. Okay, like they form alpha helices and beta sheets. If they form sheets, then we call them as beta sheets. If they are helical in nature, they are called alpha helix. So these alpha helix and beta sheets together, they again form a three-dimensional structure, which is called as globular proteins. So not all three-dimensional proteins are globular proteins. So just proteins assume three-dimensional structure. And in that again, we have different types. So in that, globular proteins are one type. Okay, this is the tertiary structure. Then the next level is two or more proteins if they form. That is called quaternary structure. Okay, so here mainly these globular proteins are nothing but the proteins which form like a globe shape, means round in shape. Okay, so these proteins mainly form enzymes. And in nature, if we see, all enzymes are proteinaceous in nature, but only one enzyme is made up of RNA. And that RNA enzyme, RNA based enzyme is ribozyme. Okay. So, so RNA is nothing but the nucleic acid, right? So now we know like what is the composition of the enzyme. Now, what is the structure of the enzyme? What are the components which are present in that? So here we have an active site. So this active site is the main region where a substrate or a cofactor or a prosthetic group comes and binds into this active site. So that means when a substrate comes and binds to the enzyme, only then the reaction will speed up. So apart from the substrate, we can have cofactors, prosthetic groups, and what are they? I will explain. Okay. So these will come and bind and they will accelerate the reaction. So this is the site where this binding takes place. And as I said, enzymes are proteins. And suppose if the protein is of 100 amino acids, only 5 amino acids, that is 5% of the total enzyme surface, will make up a active site. Okay, that means only very small portion of the enzyme will form a pocket, which is called active site. So this active site has a specific shape. It is not, the composition of the active site is not random. That means the enzymes, as I said, like out of 100 amino acids, only 5 will form the active site. So that means not any random 5. Okay, they have very specific composition. And they also have a specific shape. Okay, so that the substrate has to come and fit. Okay, and based on the shape of the active site only, the function of the enzyme depends. So this is about the structure of the enzyme. So this active site is again divided into two: binding site and catalytic site. So binding site, the name itself indicates binding. So it is only for binding of substrate. Okay. Catalytic site is where the function actually takes place. That is the catalysis. The function is initiated at the catalytic site. Now we will look into the cofactors, substrates, etc. So cofactor is nothing but a non-protein molecule. It is not a protein molecule, but these cofactors, they are involved in carrying this function, catalytic function. And so we have 20 standard amino acids in the proteins. So that means though we have many amino acids in nature, but when we see in any organism like plant, animal, human, or microorganism or anything, only 20 amino acids are actually forming the proteins. So that's why we call them as standard amino acids. So if these standard amino acids, suppose they cannot perform any function, then these cofactors will be performing the function. So these cofactors are of two types: organic cofactor and inorganic cofactor. So in this image, you can see sometimes what happens is the protein is inactive in nature. So the blue color one is inactive protein. Then comes a cofactor, the purple color one, which will come and bind to this inactive protein or enzyme. So once the cofactor comes and binds, it will become active. Then it can perform, then it will bind to the substrate and it will perform the reaction. That means along with substrate also cofactors can come and bind. Okay. So the cofactors are of two types: inorganic cofactor and organic cofactor. So inorganic cofactors are nothing but when an inorganic molecule comes and binds, it is inorganic cofactor. Okay. So here the examples are like carbonic anhydrase. It requires zinc for its activity. And hexokinase requires magnesium. Then organic cofactors means the organic molecules, they function as the cofactors. Like pyridoxal phosphate is required for glycogen phosphorylase. Okay, this is organic molecule. Then this organic molecule, based on their binding strength, again two types. It is called as a prosthetic group when it is tightly bound to the organic cofactor. Okay, like flavins, heme group, biotin, these are organic cofactors. But if they bind tightly, then they are called a prosthetic group. If they are bound loosely, then we call them as coenzyme. Example, NAD+. Okay. Then suppose an enzyme is not having any cofactor. Okay. So then we call that enzyme as apoenzyme. Right. So that means it is like a naked enzyme. It is not having any cofactor for its activity or any coenzyme. Then we call it as apoenzyme. And at the same time, if that enzyme is complexed with any organic molecule or metal ion or any other component, then we call it as a holoenzyme or holoprotein. Okay, the name itself differs. So the name indicates whether the cofactor is associated with the protein or not. Okay, that is the difference. Then substrate. So substrate is nothing but it is a reactant in a biochemical reaction. So if the same reaction, if it is taking place in a lab, usually we call it as a reactant. But if it is taking place within the body, within the biological system, then we call it as a substrate. When the substrate binds to enzyme, it will form a complex, which is called enzyme substrate complex. And where these enzymes are synthesized. Like we all know, DNA codes for mRNA, mRNA codes for protein. And what are the processes? Transcription and translation. That is DNA to RNA, it is transcription. RNA to protein, it is translation. So same way, even the enzymes are synthesized. That is DNA codes for mRNA and mRNAs will code for the enzymes. And where does the synthesis takes place? In ribosomes. Ribosomes are the organelle, like ribosomes, endoplasmic reticulum, mitochondria. All these are the organelles in cells. So these ribosomes, which are associated with the endoplasmic reticulum. So only in those ribosomes, usually this enzyme synthesis takes place. So in endoplasmic reticulum, there are two types. One is rough endoplasmic reticulum and other is smooth endoplasmic reticulum. So smooth endoplasmic reticulum, where we don't see any ribosomes. Whereas in rough endoplasmic reticulum, there are some ribosomes which are attached or associated with the endoplasmic reticulum. So that's why the surface appears rough. So that is called rough endoplasmic reticulum. And there are some ribosomes which are freely available in the cell. Okay, so these are the different types. So on this rough endoplasmic reticulum, whatever the ribosomes are present, so they are involved in the synthesis of enzymes. And this enzyme synthesis, the information required for the synthesis of enzyme is carried by the DNA, as usual, the genetic code. Okay. So once the DNA codes for mRNA, mRNA codes for protein. So that means the amino acids, they bind together and they will form an enzyme. Okay. So these enzymes are of two types, intracellular and extracellular enzymes. So intracellular is nothing but intra means within. Cellular means cell. Within the cell, the enzymes are present. Then extracellular means, extra means outside, cellular means cells. So outside the cell. So the intracellular enzymes are nothing but they are synthesized as well as they are retained within the cell. Okay, that means the enzymes are synthesized inside the cell and they are used for that cellular purpose. Okay, so usually they are seen in cytoplasm, nucleus, mitochondria, and chloroplast. Example: oxidoreductase enzyme catalyzes biological oxidation. So biological oxidation is a reaction which takes place within the cell, like especially in mitochondria. So these enzymes are involved in within, means they are synthesized inside that cell, they are utilized in that cell itself. Then extracellular enzymes are synthesized inside the cell, but they are transported from the cell to work externally. Okay, that means, for example, in pancreas, there are some enzymes which are produced and then they are transported to duodenum to perform their function. They are extracellular. So what are the characteristics of the reaction or the characteristics of the enzymes and enzyme catalyst reactions? So the main function of enzymes is they speed up the reaction. So how they speed up the reaction? By lowering the activation energy. So what is this activation energy? Means there are reactants and there are products. So the reactants have some energy and the products will have some amount of energy. And there will be always difference in energy levels of reactants and products. Okay. So this substrate has to cross that barrier of energy to convert them into the product. Okay. So that is called as activation energy. I will explain in detail in the next slides. But this activation energy is mainly reduced by the enzyme. Therefore, the reaction will speed up. And the presence of enzyme does not affect the nature as well as property of the end product. And these enzymes are not random. You cannot expect any enzyme to perform any reaction. So they are very specific. Only some enzymes will perform some reactions, and they can convert only some types of substrates into products. And very little amount of enzymes also can accelerate the reaction. They don't require lots of amounts of enzymes. Very little quantity of enzymes also can do their best. Then enzymes are sensitive to any changes in the pH, temperature, and substrate concentration. Then turnover number. So turnover number is nothing but how many number of substrate molecules are converted to products by one enzyme molecule in 1 minute. That is called as a turnover number. So every enzyme will have a specific turnover number. So here if you see catalase enzyme has turnover number of 6 into 10 power 6 per minute. That means single molecule of catalase can convert 6 into 10 power 6 substrate molecules into product per minute. Okay, so this also varies from different enzymes will have different turnover numbers. The next is nomenclature. Nomenclature is nothing but naming system. How we name an enzyme. Okay, so basically most of the enzymes are named based on their substrate. For example, maltase is an enzyme. So maltase usually functions on maltose. So the reaction is maltose. When it reacts with water, it gives rise to 2 molecules of glucose. And this enzyme, this reaction is catalyzed by maltase. So you can know that substrate is maltose and enzyme is maltase. So this way of naming is common. Other than this, there are some names which were given long back, but they are not according to this system. Okay, like pepsin, trypsin, renin, these the substrate names are nothing to do with the enzyme names. Okay, and some are like, as I said, maltose, maltase. So the suffix ase is given at the end of the name of the enzyme. Okay, like if they are involved in hydrolysis process, then it is called hydrolase. Okay, like that. These are the examples. So here there are some other examples like lactase is an enzyme which act on lactose to give glucose and galactose. Maltase, they act on maltose to form two molecules of glucose. Cellulase form on cellulose to form two molecules of glucose. Then lipase act on lipids to form glycerol and fatty acids. Then amylase act on starch. So why we are giving the name amylase here? Because the starch is made up of amylose and amylopectin. So the names amylose and amylopectin are present. That's why the name of the enzyme has come as amylase. So the products formed are maltose. Then protein. So proteases will act on proteins to give peptides and polypeptides. So these are the some of the examples. Now we will discuss about the classification of enzymes. So enzymes are classified based on the rules and regulations given by the International Enzyme Commission. So this classification is based on the type of reaction. So what type of reaction in which these enzymes are involved? So based on that, the naming is given. So there are 6 classes of enzymes. Though they have subclasses and subclasses and everything will have a numbering, but just remember the basic 6 classes of the enzymes. So that is important. So here the 6 classes are, first one is oxidoreductase, where the name, you always can expect the type of reaction based on the name. You should know the like how that name is formed. Okay, so oxido means oxidation, reductases, so reduction. So oxidation and reduction reactions are carried out by oxidoreductases. So examples, lactate dehydrogenase is the example. Then transferase, transfer, okay, name itself. So that means they transfer or move chemical groups from one molecule to another molecule. So example, hexokinase. Then hydrolases. Hydrolases are involved in hydrolysis process. So what is hydrolysis? When a, suppose example is maltose reacts with water to give two molecules of glucose. So here what is happening? Maltose is breaking down into two molecules. But here along with that, this water molecules also, it breaks down into H and OH, and H group will go and bind to the one molecule of the product which is formed from the maltose, and OH will go and add to the another molecule, so that they finally form two glucose molecules. So got it? That means suppose A plus B is equal to C. That means A is broken down into two. B is water here. So it is also broken down into two, H and OH. Now this H will go and bind to the part 1 of the A, and OH will go and bind to the part 2 of the A, so that it will form two molecules of the product. So this is hydrolysis. Hydro means water. So in such type of reactions, usually the enzymes which perform such type of reactions, we call them as hydrolases. So example, lysozyme. Then lyases. Lyase is also doing the same function, but here non-hydrolytic bond. That means the water is not involved here. Example, fumarase. Then isomerases. Isomerases are nothing but the isomers. So isomers are, they are having same chemical formula, but their structure or configuration differs. Usually in carbohydrates, we see L isomers, D isomers. That means the main, suppose glucose is there, C6H12O6. So that is the formula. And another molecules like even fructose also will have C6H12O6. But the shape means the arrangement of the groups will vary, right? So such type of groups are called isomers. So this transfer of groups between isomers is called as isomerization, sorry. And the enzymes involved are isomerases. So example, triose phosphate isomerase. Okay. Then ligases. Ligation means forming of bonds, new covalent bonds between the substrates. And formation of bonds requires energy. And this energy is derived from the ATP hydrolysis. ATP is the energy packet of the cell. So it will give the energy. So when ATP is broken down, energy is released, and that energy is utilized for the synthesis of new bonds. So such type of reactions are carried out by ligases, enzymes. Example, RNA polymerase. So what is the mechanism of enzyme action? So the mechanism can be explained in two viewpoints, two perspectives. One is thermodynamic changes and other is processes at active site. So thermodynamic changes are nothing but it is the changes in the temperature. So now all chemical reactions, in every chemical, whether it is biological or chemical, any reaction, there is an energy barrier, as I said, that is the difference in energy levels are always present. And this barrier is called as activational barrier. Okay. So here if you see graph clearly, the first reaction, that is a green color peak, if you see. So there are reactants, and from reactants to convert into product, they require lot of energy. Means these reactant has to cross all that peak, it has to go top, that much energy it has to reach, and then only it can convert into product. When it is converted into product, the energy levels again drops. Okay, means first it has to take up that much energy to convert itself into product. So this green color one is uncatalyzed reaction. That means we are not added any catalyst, whether it's a chemical reaction or biochemical reaction. No catalyst is involved. So here what happens? The substrate requires lot of energy to convert into product. Whereas in the second reaction, you can see catalyzed reaction, the red color peak, it is almost half. The activation energy which is required for the conversion of substrate into product is only half. So this can take place very easily compared to the first type. So therefore, the time will reduce and the reaction can take place very fast. So here, only a few substrate can cross the activation barrier and change into product. So that's why uncatalyzed reactions are slow in nature. Then, whereas these enzymes, they provide an alternate pathway for conversion of substrates into products. Okay. So what happens here? These enzymes, they will help in forming a transitional state. So that means where the energy required for conversion of substrate into product is reduced. Hence, the rate of reaction will increase many folds and it can happen very quickly. Okay, so this is nothing but the mechanism involved in the enzyme catalyzed reactions. So for your understanding, in a very simple way, if you see, so here you can see without enzyme and with enzyme. For example, lactose has to convert into glucose and galactose. So here lactase enzyme. Suppose this man who is performing the action, he is lactase. Okay. So now he has to move this lactose into, convert the lactose into glucose and galactose, and the peak of the mountain is very high. So what happens if the peak of mountain is very high? To climb that mountain, it requires lot of energy, lot of time, lot of efforts. So that is the activation energy. We have to get activated to reach that peak, right? So then it will drop. Please mute yourself. Okay. Then, whereas in the second reaction, if we see, here the peak is very low, right? So it will take very less time to convert this lactose into glucose and galactose. So the efforts which the enzyme is keeping is reduced, and even substrates also, right? So this is the main difference. Now the processes at the active site. So at the active site, 4 types of reaction can take place. One is covalent catalysis, acid-base catalysis, catalysis by proximity, and catalysis by strain. In the covalent catalysis, so we all know different types of bonds are present, like covalent bond, ionic bond, hydrogen bond, etc. So these covalent bonds are nothing but when an enzyme is linked to a substrate, it will form enzyme substrate complex. So here in this image, if you can see, the first step, the enzyme, which is green one, is binding with the substrate, yellow, orange. And here the bond which is formed is covalent bond. So tightly bound and it is strong. Then it will form enzyme substrate complex. And finally, the reaction takes place and the product is formed. Here the C is the one product and AB is the one product. If you see here, substrate A is one substrate and BC is another substrate, right? So this type of reaction or catalysis is called as covalent catalysis. Second is acid-base catalysis. So here oxidoreductase enzymes are involved. So here acid and base reaction. So H plus and OH minus. So usually H plus ions, when they are present, they form acidic condition. If they are less in number, then it will form basic condition. So here these oxidoreductase enzymes are involved. So in this active site, usually histidine is present, and this histidine acts as both proton donor and proton acceptor. Okay, so this histidine, it will accept or it will donate H plus ions to the substrate to form product. This type of reaction is taken place in the acid-base catalysis. Then catalysis by proximity. Proximity is distance. So when the substrate comes in close contact with the active site, so high substrate concentration is produced in the active site region, that too at the ideal position, ideal distance, okay, to convert them into the product. Then bond strain. Bond strain is nothing but, especially this is by lyases, the enzyme substrate binding, it causes reorientation of the structure. The site of active site, it will reorient itself and it will form a transitional state. That means the strained condition will take place. That is, conformational change is formed, and here the unstable condition is formed, so that the product has to remove. Okay, if it is tightly bound, the product cannot be removed, right? So first it will form a stable complex, reaction takes place, then it will become unstable, so that the product which is formed is released. Okay, these are the different types of reactions takes place in the active site. Then we have different models to explain the structure, how the association of enzyme and substrate. So first is lock and key model. It is a very popular model proposed by Emil Fischer in 1894. So this is called lock and key hypothesis. So according to this, the shape of the enzyme is rigid. Rigid means it cannot change. So as we see a specific key fits into the lock, it can open a lock. So in the same way, specific substrate or specific enzyme is associated with the specific substrate. Though it is very popular, it has its own limitation. So because of that, another model was proposed. This is called induced fit model. So this was proposed by Daniel Koshland in 1958. So this, according to this model, whenever an enzyme is exposed to a substrate, this enzyme changes its active site, means the shape of the active site, and it so that it can bind to the substrate. That means the complementarity is maintained, but it is not rigid, it is flexible. Okay. So here, this is the induced fit model. Now the next topic is enzyme kinetics. So it is a branch of science where the rate of enzyme catalyzed reactions are studied. So here the number and order of individual steps which are involved in enzyme catalyzed reactions, they are studied. And this is mainly to understand the mechanism, the role in metabolism, and how this reaction is controlled, and how a drug or an agonist or antagonist is inhibiting the enzyme. All these we can know only by knowing the enzyme kinetics. Now, so what are the factors which are involved in rate of reaction? So how rate of reaction is dependent on some factors. So what are they? One is activation energy. As I told you, it is the least amount of energy needed for a chemical reaction to take place. So here enzyme as a catalyst, it will reduce the activation energy to speed up the reaction. So here example, carbonic anhydrase. So this catalyzes hydration of carbon dioxide. So 10 power 6 carbon dioxide molecules are converted to product per second. This is very much faster, that is 10 to the power of 7 times faster than the spontaneous reaction. It is not that if there is no enzyme, no reaction takes place. No, reaction can happen, but it will be very slow. Okay. So this is the same like whatever the graph I have explained you earlier. So initial energy state and final energy state of the product. So this barrier is covered by the activation energy, and that activation energy is lowered in case of enzyme catalyzed reaction. So in the enzyme catalysis, it will increase the rate of reaction, mainly a transition state, that is intermediate state, which will form a covalent bond. That is called covalent catalysis, as I told you, but it will become unstable later. And this rate of reaction is dependent on three factors: temperature, pH, that is hydrogen ion concentration, and substrate concentration. First is temperature. So whenever the temperature is increased, the rate of enzyme catalyzed reaction also increases. But the enzymes, they function efficiently at some temperature that is called optimum temperature. For example, the enzymes, it depends on our body temperature. Okay, so suppose human beings, so the enzymes are very much stable at 35 to 45 degrees centigrade, and the temperature coefficient is nothing but it is given as a Q10 factor. That means for every 10 degrees rise in temperature, there is an increase in, that is doubling of the reaction. But it will have some level, some point where the reaction will start dropping. Why? Because the enzyme, which is a protein, it will start denature when exposed to very high temperatures, beyond their optimum temperatures. So see here the graph. At 5 degrees centigrade, it is inactive or less than 5 degrees. And 5 to 40 degrees centigrade, it is very efficient, and the rate of reaction is really high. But again, after 40 degrees, slowly it starts dropping. This is because of denaturing of the protein. Got it? Then effect of pH. So rate of almost all enzyme catalyzed reaction depends on the pH. And pH value, the optimum pH value is between 5 to 9. And high pH or low pH, both of them can ionize the enzyme and they can denature the enzyme. Okay, so because enzymes also will have some ions, so the ionization of the enzyme, denaturation of the enzyme takes place and it may lose. So that's why every enzyme will have an optimum pH. So here in the example, if you can see, pepsin has an optimum pH around 2. Then trypsin is around 7 and arginase is around 9. So like that, it will differ. The next is Michaelis Menton model and effects of substrate concentration. So there is a derivation of this equation, but no need to remember in detail. Just remember that Michaelis Menton model, it will explain you about how the concentration of substrate affect the enzyme catalyzed reaction. So here, enzyme plus substrate, it is giving rise to enzyme substrate complex. So this is catalyzed by the, yeah.
Which unit?
It is second. You don't have syllabus copy?
No, ma'am, I don't have.
Okay, later after completing the class, I'll let you know.
Okay,
Okay,
ma'am, thank you.
I think second unit also. Last topic of the second unit. Okay.
Okay.
Yeah, yeah. So this enzyme and substrate, it is giving rise to enzyme substrate complex. So this is the forward reaction. So K1 is nothing but it is the rate constant of the forward reaction. So now this enzyme substrate complex has two options. One is either it can form enzyme and product, or it can go back to enzyme substrate. That is, it can become reversible. The reaction may become reversible, or it may go forward. Okay, this again depends on the substrate concentration. So once the reaction is completed, the enzyme will become free so that it can process or it can perform another reaction. Okay. So here the equation, just remember the equation. Okay. Even that is not very much necessary, but you can just have an idea. Okay. V naught is equal to V max into substrate concentration by Km plus substrate concentration. So here V naught is the initial velocity of the reaction, initial speed. Vmax is the maximum velocity, then substrate concentration, and Km is the Michaelis constant. So how this Km has come? From the values K minus 1 plus K2 divided by K1. So what is this K1, K2, all this? From previous equation, if you see K1, K minus 1 and K2. Okay. So this equation, this is called Michaelis-Menton equation, and this mainly explains you the relation between the substrate and reaction. Okay. Here you can see how this means the relation looks like. Like our assumption is like if we increase substrate concentration, the rate of reaction increases. That is true to some extent. That means if the substrate concentration is increased, then the rate of reaction also increases. But after some time, after saturation point, though you add more and more substrate, the reaction remains standard. It cannot increase. Why? Because the active sites are limited. Even if you add more and more substrates, you cannot accommodate the substrate in the active site. Therefore, the enzyme catalyzed reaction cannot take place. Okay. So this is how the Michaelis-Menton plot looks like. Now, what is the importance? Like why we are studying this enzyme catalysis or enzyme kinetics, all these things? Because they are of pharmaceutical importance. That means usually, for example, there are some diseases like cancer or any other metabolic disorders where a particular enzyme catalyzed reaction is culprit in causing the disease. So you want to block that enzyme catalyzed reaction. Okay. So how to block that? By using some drugs. So unless you know how the reaction is taking place, well, that means which enzyme is involved, how much speed means what is the chemical reaction, rate of reaction. So what are the products, what are the activators, inhibitors, structure of the enzymes. You should know everything before developing a drug to target that particular pathway or that particular enzyme catalyzed reaction. So therefore, enzyme kinetics is a principal tool for the drug discovery. Okay. Now we have two other concepts like inhibition and activation. So inhibition is nothing but where we are stopping the enzyme catalyzed reaction. So here inhibitors are the molecules. So inhibitor is any substance that will reduce the enzyme catalyzed reaction. How? By binding to the enzyme. So when inhibitor comes and bind to the enzyme cannot perform its function. Okay. So types of inhibition. So the inhibition is of two types, reversible, irreversible. So reversible is nothing but whenever a reaction, whenever an inhibitor is added to the reaction, and suppose after some time you don't want the action of inhibitor, so you are withdrawing that inhibitor and you can again resume that reaction. That means you are stopping, and if you want you can also start again. That kind of reactions are nothing but reversible reaction. Whereas in irreversible reaction, once you stop, you cannot reactivate the reaction. Okay. So in the reversible, we have four types: competitive, uncompetitive, mixed and non-competitive. So competitive, I'll explain you in detail everything. So these are four types of reactions. So in the competitive inhibition, so the here you can see in the image the enzyme, purple color one is enzyme, and you have only one active site where either substrate or inhibitor can bind. So there is a competition between substrate and inhibitor. So only one can come. If substrate comes and binds, the reaction takes place. If inhibitor comes and binds, the reaction will stop. So this type of inhibition is nothing but the competitive inhibition. That means you should add more inhibitors than the substrate, so that definitely the inhibitor will go and bind to the active site. Okay. Example is statin drugs like Lipitor. So this will go and compete with the HMG-CoA, which is a substrate, which will go and bind to the HMG-CoA reductase. This is mainly in the cholesterol. If any person is having more cholesterol, so for them, they will give statin drugs. So this is the mechanism of action. Then uncompetitive inhibition. Here, the enzyme will have two different sites for binding of substrate and inhibitor molecules. So here, that means there is no competition. Substrate goes and binds to the active site, and inhibitor goes and binds to the another site, which is called allosteric site. So example here are mainly drugs involved in methanol poisoning or ethylene glycol poisoning. And here, mainly the inhibitors are tetramethylene sulfoxide, 3-butyl thiolene 1 oxide. So these two are the drugs. Then mixed inhibition. In this you can see enzyme inhibitor, then enzyme substrate inhibitor complex. It is also same type of like non-competitive inhibition. Okay. Then irreversible inhibition, where there is a covalent attachment between the enzyme and inhibitor, so that the enzyme will completely lost its catalytic activity. Okay. So therefore, once it is treated, it cannot reverse. Example, aspirin. So here it targets and modifies the enzyme involved in the inflammation. Then suicide inhibition. Suicide inhibition is, it's an unusual type where irreversible inhibition takes place, and this inhibitor, the enzyme converts the inhibitor into a reactive form. Okay, so that means it will become very much active and it will come and bind to the active side. That means the enzyme itself is driving the inhibition of its own catalysis. Okay, then activation. So activation is nothing but where we are increasing the catalysis of enzymes. So here there are two types of activation. One is by cofactors and other is by converting the enzyme precursors. So first one is by cofactors. So here we have various cofactors. Already I told you what are cofactors. So here they will increase the enzyme catalyzed reaction efficiency. Okay, like magnesium is required for DNA polymerase enzyme. Then zinc is required for horse liver deoxygenase enzyme, dehydrogenase enzyme. Okay. Then conversion of enzyme precursor is nothing but, so here proteolysis of a particular enzyme will convert it into other product. Means trypsinogen is an inactive form of the enzyme. It is an inactive form of the enzyme. It is a protein, of course. So this trypsinogen will get converted to trypsin. So here you can see by zymogens. So here you can see the trypsinogen is converted to trypsin by enteropeptidase. Another enzyme will come and make them active from inactive form. In the same way, trypsin will activate proelastase to elastase. Then chymotrypsinogen to chymotrypsin. Then procarboxypeptidase to carboxypeptidase. Prolipase to lipase, like that. Okay. The next is enzyme specificity. So enzymes are highly specific in nature. So they can function only on particular type of substrate. So example, oxidoreductase cannot perform hydrolase reactions. Okay, like that. So the specificity of different types: bond specificity, group specificity, absolute specificity, optical or stereo specificity, and dual specificity. So bond specificity means some enzymes can perform only on the molecules which are having a specific bond. Like amylase can act only on alpha 1,4 glycosidic bond. Okay. Then group specificity. That means the enzymes can perform only on specific groups. Like some can perform only on the amino groups. Some perform only on the aldolases, some on ketosis, like that. Then substrate specificity, they will have very specific substrate. Like maltase act only on maltose. Uricase act on uric acid. Okay. Then optical or stereo specificity. Here, optical activity or stereochemistry or optical configuration is nothing but the how the groups are arranged. So usually in carbohydrates or amino acids, you can see the arrangement of groups in the space. Like they are based on that, they will give D and L name, D amino acid, L amino acid. Okay, so some enzymes, when we discuss about proteins and all that, we will discuss in detail. But here, some enzymes act on only D amino acids. Some act on only L amino acids. Okay, like that. This is called optical specificity. Then dual specificity. Here, there are two types of dual specificity. That means same enzyme can act on substrate by two different reactions. Okay, so here mainly isocitrate dehydrogenase. It acts on isocitrate by oxidation and decarboxylation by another reaction. Is same enzyme but two different substrates. And sometimes two substrate with only single reaction. That means here you can see xanthan oxidase acts on xanthan, hypoxanthin, xanthan and hypoxanthin. Both are the substrates in the same reaction. That means two steps are catalyzed by the same enzyme. Got it? This is another type of dual specificity. Okay. So these are about like specificity. So today we have discussed about, so what is enzymes? What do you mean by enzyme? Okay. What are the mechanism of action, synthesis, catalysis? Okay. So what are the factors affecting activation, inhibition, specificity, all these concepts. Okay. First, let me stop the screen sharing. Then I will meanwhile, if you have any doubts, you can ask
Excuse
me.
me, ma'am. Please
Yeah.
share the syllabus.
So here, just a second. Do you have any group or some you don't receive like book? You don't have books?
We have the books, but our subject is principle of aquaculture, zoology.
You are not fifth semester?
We belongs to fifth semester only, ma'am.
Okay. So in this you have this no animal physiology and biochemistry.
No, ma'am.
Fifth semester English medium only, no?
Yes, ma'am.
So what is the syllabus? What is your syllabus? Which subject are you studying now?
Our syllabus is principle of aquaculture, ma'am.
But
Discipline
I have
specific elective course.
not, this is not elective, this is core.
Okay, ma'am. Physiology and biochemistry.
Yeah.
Okay.
Okay, in this unit 9, enzymes classification, inhibition and regulation. So that topic I have covered.
Okay, ma'am. Ma'am,
Okay, so from unit, yeah.
can you give us important questions also?
Once I finish, I'll give in the last topic. Okay.
Oh, okay, ma'am. Sure.
First, let me
Thank
know
you.
like how many classes it takes, like all these things. So
Yes, ma'am.
from unit 9 onwards, you have biomolecules. So first, let me cover the biomolecules so that
Okay.
you can understand the physiology. Okay, whatever maximum topics I'll try to cover. Okay.
Yes,
So
ma'am.
if you have any doubts, you can ask. You can go through the book once again. Meanwhile, before next class. And if you have any doubts, you can ask. Okay.
Okay, ma'am.
Okay, ma'am. Thank you. Thank you. Thank you so much.
Okay. Okay. Yeah, thank you so much.